Hello students, welcome to EPG Pathsala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about module on a proton motive force under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We will try to understand the function of proton motive force in the production of adenosine triphosphate. We will also try to examine the importance of a proton gradients power the synthesis of ATP. We will examine the importance of shuttles for aerobic oxidation of cytosolic NADH, and we will try to understand the cellular respiration, which is regulated by the need for ATP. It shows the concept map of proton motive force. This figure gives the overview of general use of proton gradients in the proton motive force. We use in NADPH and ATP synthesis, heat production, flagellar rotation, electron potential, and active transport. A proton motive force is energy that is generated by the transfer of protons or electrons across an energy transducing membrane and can be used for chemical, osmotic, or mechanical work. It can be generated by a variety of phenomena, including the operation of an electron transport chain, illumination of a purple membrane, and the hydrolysis of ATP by a proton ATPase. Necessity for proton gradient. The proton gradients are required in the boils back down to chemistry. In other words, to convert carbon dioxide into organic molecules, life attaches hydrogen atoms to carbon dioxide. This pathway is exothermic, that is releasing energy that can be captured as ATP right through the pyruvate, one of the central molecule in cell metabolism. But there is a problem pointed out by William Martin, who collaboration with Russell. All cells that use the acetyl coenzyme A pathway today depend on proton gradient. None of them can grow by fermentation because carbon dioxide is a stable molecule and does not react easily even with hydrogen. Carbon dioxide is a bit like oxygen in this respect. Once it starts to react, it's not easy to stop. Cells need the equivalent of a spark to get carbon dioxide to react, and for cells, that spark is ATP. The problem is that the reaction of carbon dioxide with hydrogen release energy, but not a lot, only enough to make 1 ATP. That means cell have to spend 1 ATP to gain 1 ATP. So there is no net gain. If there is no gain, there is no growth. No growth, no life. Gradients break that cycle. It's not quite true to say that the reaction of carbon dioxide with hydrogen release enough energy to make 1 ATP. It is actually enough to make 1.5 ATPs. But of course, there is no such things as 1.5 ATPs. at least not by stoichiometric chemistry. So the spare energy from the reaction as chemistry is lost, but that doesn't happen with a gradient. In principle, a reaction can be repeated over and over again just to pump to a proton over a membrane. When enough protons have accumulated, the proton motive force powers the formation of ATP. So, a gradient allows cell to save up proton as loose change, and that makes all different in world the difference between growth and no growth, life and no life. Coupled nature of respiration in mitochondria. Oxygen consumed by only with ADP in case of excess of inorganic phosphate. First oxidation of substrate is coupled to the phosphorylation of ADP. When ADP is present, then respiration, that is consumption of oxygen, proceed further. Only the amount of oxygen consumed depends upon the amount of ADP added. Uncoupled DNP, that is oxygen consumed with ADP. Uncoupler are lipid soluble weak acids. It stimulates the oxidation of substrate in the absence of ADP. Both acidic and basic forms can cross the inner mitochondrial membrane. Uncouplers deplete any proton gradient by transporting proton across the membrane. First we require, if you see the Michler's postulate for chemiosmotic theory, first we require intact inner mitochondrial membrane for maintenance of a proton gradient. Electron is transported through the electron transport chain for the generation of a proton gradient. The membrane spanning enzyme ATP synthase catalyze the phosphorylation of ADP in a reaction driven by movement of proton across the inner membrane into the matrix. Cytosolic NADH must enter in the mitochondria to fuel oxidative phosphorylation, but NADH and NAD plus cannot diffuse across their inner mitochondrial membrane. Thus, two subtle systems we used in the reducing equivalence. These are first Glycerol phosphate shuttle, dominate in insect flight muscles, and second is Malate-Aspartate Shuttle, which predominant in liver and other mammalian tissue. Chemiosmosis is the movement of ion across a selectively permeable membrane down their electrochemical gradient. More specifically, it relates to the generation of ATP by movement of hydrogen ion across a membrane during cellular respiration or photosynthesis. Hydrogen ions, that is in form of protons, will diffuse from an area of higher proton concentration to an area of lower proton concentration, and an electrochemical concentration gradient of protons across a membrane can be harnessed to make adenosine triphosphate. This process is related to osmosis, the diffusion of water across a membrane, which is why it is called chemiosmosis. Adenosine triphosphate synthase is the enzyme that make adenosine triphosphate by chemiosmosis. It allows proton to pass through the membrane and use the kinetics energy to phosphorylate adenosine diphosphate, making adenosine triphosphate. The generation of adenosine triphosphate by chemiosmosis occurs in chloroplast as well as in most bacteria and archaea. A difference of soluble concentration between two compartments separated by a biological membrane generates a tendency to equilibrium, which in case of proton is called proton motive force. This is measured in terms of potential energy resulting from the difference in concentrations between the two compartments. With a charged solute, the potential has two components: the chemical that is concentration, and the electrical potential. That's why it is named electrochemical potential. Figure shows glycerol phosphate shuttle. The glycerol 3-phosphate shuttle is a mechanism that generates ADP plus from NADH, a byproduct of glycolysis. It's important in transporting a reducing equivalent is secondary to the Malate-Aspartate Shuttle. Mechanism is that in this shuttle, the enzyme glycerol 3-phosphate dehydrogenase 1 convert dihydroxyacetone phosphate to glycerol 3-phosphate by oxidizing one molecule of NADH to NAD+. Glycerol 3-phosphate gets converted back to dihydroxyacetone phosphate by an inner membrane bound mitochondrial glycerol 3-phosphate dehydrogenase. At this time, reducing one molecule of enzyme bound to flavin adenine dinucleotide, that is FAD, and converted into FADH2. FADH2 then reduce coenzyme Q ubiquinone or to ubiquinol, which enters in oxidative phosphorylation. This reaction is irreversible. The glycerol 3-phosphate shuttle allow NADH synthesized in the cytosol by glycolysis to contribute to the oxidative phosphorylation pathway in the mitochondria to generate ATP. It has been found in animal, fungi, and in plant. The following figure shows the Malate-Aspartate Shuttle. The Malate-Aspartate Shuttle is a biochemical system for translocating electron produced during glycolysis across the semi-permeable inner membrane of the mitochondria for oxidative phosphorylation in eukaryotes. These electrons enters in the electron transport chain of the mitochondria via reduction equivalent to generate ATP. If you see the mechanism of Malate-Aspartate Shuttle, the primary enzyme in the Malate-Aspartate Shuttle is malate dehydrogenase. First, in the cytosol, malate dehydrogenase catalyzes the reaction of oxaloacetate and NADH to produce malate and NAD+. Once malate is formed, the first antiporter, that is malate alpha ketoglutarate, import the malate from the cytosol into mitochondrial matrix and also export alpha ketoglutarate from the matrix into the cytosol simultaneously. Oxaloacetate is then transformed into aspartate by mitochondrial aspartate aminotransferase. Since aspartate is an amino acid, an amino radical needs to be added to the oxaloacetate. The second antiporter, that is the glutamate aspartate antiporter, imports glutamate from the cytosol into matrix and export aspartate from the matrix to the cytosol. The net effect of the Malate-Aspartate Shuttle is purely redox. NADH in the cytosol is oxidized to NADH and NADH in the matrix reduced to NADH. Since The Malate-Aspartate Shuttle regenerates NADH inside the mitochondrial matrix. It is capable of maximizing numbers of ATPs produced in glycolysis, ultimately resulting in a net gain of 38 ATP molecule per molecules of glucose metabolized. There are three active sites for the functioning of ATP synthase. One in each beta subunit. The sigma unit form a rotor. There are three units altogether form a rotor. Rotation of the gamma subunit inside the alpha 3 beta 3 hexamer cause domain movement in the beta subunit, opening the and closing of active site. The alpha beta gamma alpha 3 beta 3 unit is the starter. The F0 channel is attached to the alpha 3 beta 3 by the alpha beta gamma arm. F0 F1 ATP synthase use the proton gradient energy for the synthesis of ATP. An F-type ATPase which generate ATP composed of a knob and stalk structure. F1 knob contains the catalytic subunit and F0 stalk has a proton channel which spans the membrane. Passage of the proton through the F0 that is stalk into the matrix is coupled to ATP formation. Estimated passage of proton and ATP synthesized. Passage of the proton through F0 that is stalk channel cause the rotor to spin in one direction and the starter to spin in the opposite direction. Conformational change in the ATP synthase. First ADP inorganic pyrophosphate bind to an open site, inward passage of proton, conformation change, ATP synthesis from ADP and pyrophosphate, ATP release from open site, ADP and pyrophosphate form ATP in the tight site. In most cases, the proton motive force is generated by an electron transport chain, which act as a proton pump using the energy of electron from an electron carrier, that is gives free energy of redox reaction to pump proton, that is hydrogen ion, out across the membrane, separating the charge across the membrane. In mitochondria, energy released by the electron transport chain is used to move proton from the mitochondrial matrix to the intermembrane space of the mitochondrion. Moving the proton out of the mitochondrion create a lower concentration of positively charged proton inside it, resulting in a slight negative charge on the inside of the membrane. In mitochondria, the proton motive force is almost entirely made up of electrical component, but in chloroplast, the proton motive force is made up of mostly of the pH gradient because the charge of protons is neutralized by movement of chloride and other anion. In either case, the proton motive force needs to be around 50 kJ per mole for adenosine triphosphate synthase to be able to make adenosine triphosphate. It is generally accepted that the chemistry of water was the most crucial determination in shaping life on Earth. Among the more important chemical feature of water is its dissociation into proton and hydroxyl ion. The presence of relatively high proton concentrations in the ambient solution resulted in the evolution of proton pumps during the dawn of the life on Earth. These proton pumps maintained neutral pH inside the cell and generated electrochemical gradient of proton, that is proton motive force, across their membrane. The existence of proton motive force enabled the evolution of portals driven by it that are most probably among the most primitive portals in the world. The directionality of the substrate transported by the portal could be to both sides of the membrane because they can serve as a proton symporter or antiporter. So students, let us summarize what we have learned in this module. Bacteria and archaea also use chemiosmosis to generate adenosine triphosphate. Cyanobacteria, green sulfur bacteria, and purple bacteria create energy by a process called photophosphorylation. These bacteria use energy of light to create a proton gradient using a photosynthetic electron transport chain. Non-photosynthetic bacteria such as E. coli also contain ATP synthase. In fact, mitochondria and chloroplast are believed to have been formed when early eukaryote cell ingested bacteria that would transfer energy using chemiosmosis. This is called the endosymbiotic theory. Chemiosmotic phosphorylation is the third pathway that produces adenosine triphosphate from inorganic phosphate and an ADP molecule. This process is a part of oxidative phosphorylation. The proton motive force occurs when the cell membrane becomes energized due to electron transport reaction by the electron carrier embedded in it. Basically, this causes the cell to act like a tiny battery. Its energy can either be used right away to do work like power flagella or be stored for later in adenosine triphosphate. So student, this is all about proton motive force. Thank you.